call this meeting to order. Welcome to Life in the Raccoon River Watershed, and welcome from the Raccoon River Watershed Association. My cohorts and I are the board of the Raccoon River Watershed Association, and many of them are here online and helping the Raccoon River Watershed Association, easily called the RRWA, so I don't have to say it a bazillion times, started in 2005. I haven't seen Mike Delaney. I haven't seen Mike Delaney here yet, but he probably will be with us. And he's one of the founders and still very, very active. And then some of my co-hosts are also board members: Christine Curry, Ben Maas, Robin Fortney is out there, Karen Kelleher, and so forth. Bob Rye. We are happy to do an annual event. This one, I think, is something like maybe our 10th or 11th. But this is the first time we've ever tried to do it virtually, and we're excited that we can bring such great speakers to you in such a virtual way that none of us have to leave our living rooms to hear and see Chris Jones and Paul Weihe and Peter Levi, Adam Dixon, and Sylvia Secchi. I'm just really excited. Ben's going to take it from here and start us off. Christine and Bud are going to make sure that everybody gets their questions in and gets them recognized. So if you have questions for any particular speaker, type them in the chat, and we'll get them after each speaker is done. We'll have a little time at the end if you want to stay around and chat with each other. We'll take a break somewhere in the middle there, and Ben will help. We'll be doing some screen shares here in a minute, and we're just really, really happy that you all could join us. Ben, are you up there?
Yep, I'm here. So again, thank you all for joining us, giving up some of your time on Saturday to learn about the going-ons of Iowa. Between each speaker, we'll have just a little bit of a break for transition, and then we'll have a formal break after Paul's talk in between Peter. And then, as Chris mentioned, there will be opportunities for Q&A. Please, just to keep things somewhat organized, we'll be monitoring the chat window. Please type your question in there for us. Up first is Chris with the University of Iowa and Hydrosciences and Engineering at IIHR. He will be talking to us about Iowa stream nitrate and a 2020 update on that, on their longer term project. So Chris, whenever you are ready, the floor is yours.
Okay, sorry about that. I was muted. Thanks for having me. Appreciate the opportunity to present here today. You know, I've presented to this group many times, and so happy to be here once again. And so, as Ben said, I work with IIHR-Hydroscience and Engineering at the University of Iowa. And if you know Iowa City, this building, the Stanley Building, houses IIHR. The Institute has been at the university for exactly 100 years. The early research focused on ship hydrodynamics and dam design and hydraulics. And so it's relatively recently that the Institute has committed some resources to studying water quality. And so that's when I came over in 2015 from the Iowa Soybean Association. And so this is where we are. There's about 50 Ph.D. Scientists and faculty affiliated with the Institute, and about 100 graduate students. And these grad students are from all over the world. I do have an active website. I invite you to visit it if you're interested in what we do. I do post all my slides. It usually takes me a week or two to get them up there. And you feel free to use the slides if you want to use them. And so I also write blog posts there. I try to post something every couple weeks or so. And if you're interested in agriculture and Iowa water quality, I invite you to check that out. The main purpose of my job is to oversee the water quality sensor network that's run out of IIHR and funded largely through the Iowa Nutrient Research Center at Iowa State. And so this is one of the sites. We have 2 field people that deploy and maintain the system, and they work really hard to keep the whole thing going. And this is a site, I think this is on Clear Creek near Iowa City. There's about 70 sites around the state of Iowa where we measure water quality continuously. The blue sites are what I call the IIHR sites, or ones that are funded through the Nutrient Center at Ames. And we also harvest data from about a dozen USGS sites, and those are shown in green. We also have about a dozen sites funded through the Iowa Watershed Approach Project, which is the funding from Housing and Urban Development. And so at all the sites, we measure nitrate, nitrogen. At a subset of sites, we're measuring these other parameters shown on the left. And so this is a site at the Wapsipinicon River at DeWitt, Iowa. And so the sensors slide down through that PVC pipe shown on the left, and they're immersed in the moving river water. Up on the bank, we have a dry box with a solar panel. Powers a deep cycle battery. There's a data logger in the dry box, along with a modem that sends data back to Iowa City in 15-minute packets. And so this is a site near the town of Osage, Iowa, northeast Iowa, measuring the water going in and the water leaving, constructed wetland. It's actually the second largest constructed wetland in Iowa. We've had 2 students get graduate degrees studying how this wetland sequesters and processes nutrients. And so we have one of the sites here is at the Raccoon River at Van Meter, and this was the very first real-time nitrate sensor deployed in a stream in the United States. And I believe, if I remember correctly, it's been a while ago, Raccoon River Watershed Association helped us with this a little bit financially to get this deployed. And so this is when I worked at the Waterworks. I think this was deployed in 2006. And so you can see it there on the Raccoon River, and it's still there. USGS runs it now. So all this data streams into this website, the Iowa Water Quality Information System. This is really a fantastic tool. I really encourage you to go there and take a look at this thing if you haven't already. If you get to this page, all the sites are color coded by the nitrate concentration at the present time. And so you click on a site, it gives you the nitrate concentration right now, and then it delineates the upstream area from the sensor. And so there's a lot of information you can get from the site. I could spend a whole presentation just navigating the site for you. So I just would encourage you to take a look at this. An enormous amount of data streams in through the network. As you can imagine, 70 sites. We're getting data every 15 minutes. And so I work closely with the state geologist, Keith Schilling, to try to publish, research related to the data and see, these are some of the topics that we've published papers on over the last 5 or 6 years. And so I need to update this. Actually, there's quite a bit more to add to this. And so Iowa, there's a lot to talk about here. It's the reason we have things to study. We have this, intense agricultural system and all the impacts on water quality that we are interested in. And so we know that our water quality here is less than what we would like it to be. And why is that? Well, these maps on the right, very famous map here of a farm in Winnebago County in northern Iowa. And we can see in 1941, we had this real diversity of land uses. We had some corn, but, an awful lot of oats, hay, and a lot of pasture. And then we fast forward to 1976 and, all that stuff is gone except for the corn. And then we've added in soybeans. And so when soybeans came in the 50s and 60s, that displaced all these other land uses. And so consequently, we have a real lack of diversity on the landscape that contributes to our nutrient problems. And then we add in all these animals. We've got 20, at any one time, we have 25 million hogs and 80 million chickens, and then these others. And so this is a tremendous burden on our streams and lakes and our environment in general. And so now this is what our farming system looks like. And so we have these nice clean fields of GMO corn and soybeans you see there at the top. We have a huge hog industry. And, if you didn't know any better, you wouldn't even know that there are hogs living in Iowa because they're all, inside these barns there in the middle. And we never see them. These hogs never see the light of day. And then, of course, we have the ethanol industry there at the bottom. And so this is all just a really leaky system. We have no diversity here. And so the system has no capacity to sequester nutrients and other pollution. And so this produces, this green water that we see. And so this is a clear plastic vessel. I blocked out some names that were on a label here. This was the Des Moines River last summer. And so this is the green water. This is a drinking water source for Des Moines. And so this is what many of our lakes and streams look like in the summer. And I think we all know this, and, what can we do about it? And so when we look at water quality here for the last year, I thought I would kind of present some recent data for folks and they could see what the current situation is. These are the watersheds that I use to quantify nitrate loading for the state of Iowa. And so some of the sub watersheds there I list in the bigger ones that you might recognize. And so these cover approximately 78% of the state, of the water leaving the state. We have sensors, nitrate sensors at the outlets at each of these watersheds and that are measuring nitrate concentrations. And then we have discharge measurements from USGS. And so when we look at 2020, we look at the land draining to the east, to the Mississippi River, east of the purple line there. Last year, about 435 million pounds of nitrogen fertilizer, nitrate nitrogen left the state in those watersheds. And so we had a nitrogen loss per crop acre in that part of the state of 27 pounds per acre. Going to the Missouri River, we have a nitrate load of 191 million pounds in 2020, a nitrate loss per acre of about 23 pounds per acre there. And so what we see generally is a higher concentration of nitrate in the western streams. And so there's more nitrate per unit volume of water, but the loads or the total mass of the nitrogen leaving the state in those watersheds tends to be less because it's drier in western Iowa. And so we can see total runoff out there was 10 inches last year. Total runoff going to the Mississippi was 13 inches. These show loss on a watershed basis. And so the black number or top is the loss in pounds per total acre of the watershed. The red is the loss in pounds per crop acre, and the blue is the runoff. And so some things to look at with this map, we see Northeast Iowa, we really have just a tremendous loss per crop acre. We can see here the Yellow River, 78 pounds per crop acre. And so we don't have a lot, or we don't have a really high density of crop production up here. Only about half the land is cropped, but we have a tremendous amount of nitrate loss from that area, from those crop fields, because of the geology there, thin topsoils, the karst geology transfers the water to the stream network very rapidly. Then also you might note up here, Northwest Iowa, the Floyd River, 36 pounds per crop acre, but very dry, less than 10 inches of runoff. And we look at this, and we have 36 pounds per crop acre. Well, that's the same as the Wapsie. Look at the Wapsie is about 35 pounds per crop acre, but the runoff was double in the Wapsie. And so we see much higher concentrations of nitrate in western Iowa, because especially up here, because of real high density of livestock. So we have a tremendous number of hogs. The Floyd River is about 900 square miles, and there's about 900 hog confinements. And so there's essentially one hog confinement per section up there. And so this contributes to stream nitrate loading. And so when we look at this within the context of the larger system, the Mississippi River Basin, which we can see there, this is Iowa draining to the Missouri River over time. And so this is our fraction, or our percentage. And so the Missouri River watershed is huge, right? It goes all the way to the Continental Divide and Yellowstone Park. We have 3.3% of the land in the Missouri watershed. We contribute 12% of the water, but we contribute 55% of the nitrate. And so you can see in some years the Missouri River would have almost no nitrate were it not for the contributions from Iowa. And so we see here 2006, almost 90% of the nitrate in the Missouri was from Iowa. The upper Mississippi, we can think of this as the Mississippi watershed upstream of the Missouri confluence. We have 21% of the land there in the red line. We have 21% of the water and 45% of the nitrate in that system comes from Iowa. And then down at the Gulf of Mexico, the red line again, we have 4.5% of the land. We contribute about 6% of the water and about 29% of the nitrate. And so we can really see how, as Todd Dorman at the Cedar Rapids paper said, the dead zone starts here. This is where it starts. And if, we're going to try to, halt this dead zone down in the Gulf of Mexico, we really need to get serious about what we're doing up here. And so how much nitrogen leaves Iowa? Well, right now, the 5-year running annual average is about 420,000 tons. And when we look at that 5-year running annual average, that's about doubled since 2003. The data that we're using goes back to 1999, and the first year I can calculate that is that 5-year average is 2003. So that's about doubled statewide. Going to the Mississippi, it's increased 72%, and going to the Missouri River, it's almost tripled. So we can see, and so what's happening in the Missouri? Well, we've had a lot of conversion over there over the last 30 or 40 years of pasture to row crop. The cattle industry has sort of declined here, and we've taken a lot of cows off pasture, and so that land has gone into row crop and soybeans for the most part. And so then we also have a real concentration of hogs in Northwest Iowa, and I think that's part of the equation here. And so this is a little graph I made, and I take the fraction of Iowa's nitrogen load for a watershed, and I divide it by the fraction of the water that's leaving that watershed. And so like here at the top, the Floyd River, the fraction of its nitrate load is more than double the fraction of its discharge or its water. And so we can see here by that measure, all these watersheds in brown or orange are real high density livestock watersheds. And so we know now for a certainty that where we've concentrated the hogs is having a disproportionately large effect on stream nitrate. We really can see that. And so moving forward, I mean, it's abundantly clear that the hog industry is wanting to expand here in Iowa. And so, you know, can we reconcile our water quality objectives with the economic objectives related to the livestock industry and especially hogs? And I would say no. And so this graph, a very common way we look at things on the x-axis here, we have the fraction of a watershed that's in row crop. On the y-axis here, we have the nitrate concentration. Each one of these dots is a watershed. And so, you know, this has a real solid pattern here, which we've known for a long time, where these watersheds, you can basically multiply its row crop fraction by 10 and get what the nitrate concentration is going to be. But we have these outliers, you know, like I said, the Floyd, South Fork watershed in north central Iowa, Rock River, where we have real high density of livestock. And then we have northeast Iowa here, where the karst geology makes nitrate loss really almost inevitable. And so this gives us some ideas of what we can do, where we can target our efforts. And so I always say we have a mass balance problem here in Iowa. We have a really hard time coming to grips with that. The wide bars here are the inputs. And so the dark green is a commercial fertilizer. The orange is a hog manure. The lighter green is fixation from soybeans and some others in there. And then the narrow yellow bar is the amount of nitrogen that's leaving the state in the crop. And so we can see how the inputs are so substantially larger than the outputs, right? And so we have this surplus. And I always say, you know, that surplus, it's not going into the Milky Way. Something's going to happen to it. And, you know, 32% of it makes its way into our streams. And so we just cannot come to grips with this idea that we have this mass balance problem on the landscape where our inputs are, you know, so much larger than what is removed in the grain. And so, you know, a lot of the discussion these days is about soil health. And, you know, somebody asked me once, are you the guy that said we're not going to soil health our way out of this? And yes, I say that because we just fail to deal with this mass balance issue. And so we look at this percent change since 1999. River nitrate loading has increased 83%. Our yield, so grain nitrogen, we can think of our yield is increased 27%, and our total inputs have increased 36%. So this is exactly what we would expect out of an ecological system as we try to squeeze every last bit of production out of it. You know, that's going to come at a disproportionately large environmental cost. And it's my opinion that soil health is not going to rectify this until we come to grips with this idea. You know, I think it's a fantasy to think that these measures to reward and incentivize soil health are going to improve water quality, at least in the short term. And so, you know, I point out this paper from Iowa State. You know, there's a lot of people in government and academia and the industry, obviously, that want to intensify what we're doing. And so, we have Iowa State suggesting here that we really need to increase the amount of drainage in Iowa to intensify the system. And so, we really have to ask ourselves, can we reconcile all this with what we want from our streams? So I'm going to skip that. And so this map I have on my website, and, when I convert all the fecal waste in our watersheds to human equivalents, this is the map I get. And so, for example, a hog produces as much fecal waste as 4 human beings. A beef cow, I think it's 14. A dairy cow, it's 18. And so when I convert that to a human equivalent population for all our watersheds in Iowa, this is the human population, the equivalent human population that's living in our watershed. So we have the animal equivalent of 168 million people here. Okay, and upstream of Des Moines, we have Chicago, Tokyo, Paris, Rome, Oklahoma, and Alabama. And so can we get the water quality we want by continuing to cram animals into these watersheds? And I'm saying no. So what can we do? This is my low-hanging fruit. Stop cropping in the 2-year floodplain. We've got 400,000 acres in crops that we farm in the 2-year floodplain. It's crazy. Why do we do that? Ban fall tillage. Iowa State has been telling people this is dumb since the 1980s. You can drive from Des Moines to the Minnesota border in November and see all sorts of fall tillage going on. Why are we doing this? Ban manure on snow and frozen ground. Yes, we do have some laws here, but they're riddled with loopholes, and it's really easy to get around it. Why do we let this happen? Make farmers adhere to ISU fertilization guidelines. We know that farmers are over applying both nitrogen and phosphorus. Again, this is the mass balance problem. And then finally, I think we need to reformulate our CAFO regulations. We especially need to look at those manure management plans and look at how much nutrient they're allowing farmers to apply to fields. The manure management plans give license to farmers to over apply both nitrogen and phosphorus. Until we get around that, we're going to have problems. So that's my last slide. Again, thank you for the invitation. There you have it.
Hey, Chris.
Yeah.
We do have one question so far. Let me get to it.
I sent it to you in your own.
Yeah, so it came from Matt, but with the karst topography in northeast Iowa, are there any controls or records kept? Question.
Well, there's a long data record at the Big Spring fish hatchery, and so a lot of papers have been published by that. In fact, I have a couple, and so we do know that yes, in the 80s, the groundwater in northeast Iowa had gone bad. I mean, a lot of the issues then were atrazine and the pesticides, but as that system went from these small dairies and small farms, and then tried to, do what everybody else was doing in Iowa, which was corn and soybean, 24/7. We know that the groundwater up there declined in quality for sure.
And a second part of that question that you had, I think you already kind of touched on that though. But with the flow of nitrate into groundwater, is there a greater amount of nitrate entering into municipal water supplies?
Well, Manchester, which really isn't on the plateau, but is in a karstic area, has a bad nitrate problem in their groundwater. I don't have right at the tip of my tongue water systems up there that are impaired for nitrate, other than I do know Manchester has a bad problem.
Yeah. Peter, are you raising your hand?
I did. Hi, Chris. Thanks for a great presentation. I always appreciate how you're able to put such amazingly large data into perspective, like that one with the cities and such, showing the cities that are upstream of us in terms of manure. I was curious, what are your thoughts, since Iowa seems to still be kind of behind in making significant change here? Is there anywhere in the Midwest that is doing it right or doing a really good job, if not Iowa, of course?
Well, we do know Indiana, about 11% of the land is in cover crops. We have about 4%. We know Minnesota has some regulations on fertilizer application. And so I don't know that anybody has it right, but there are places that are doing more than us. So.
Thanks.
We have one more question that popped in from Lee. What is a reasonable time to apply dry barn manure? This is the first question.
Well, the closer you get it to, when the crop needs it, is obviously the best. Again, I think we spend a lot of time on the when and not enough time on the how much. And so we've tried to sort of solve the how much problem by asking ourselves, when and how and where and all these other things, because we don't have the courage to, talk about how much. And so I'm not a big proponent that, any of these fine tuning things has had a measurable impact on water quality. Are they intuitive? Yes. There are things that are intuitive. We know things are, particularly bad. But I hate to parse it out between, the when until we come to grips with the how much.
Thank you. I have another one, another question from Mike. Is no-till or cover crops better on preventing nutrient loss?
No-till, there's not a lot of evidence that no-till reduces nitrogen loss. Cover crops, yes, on average. Over the long haul, we think will reduce nitrate loss about 30% as an average. Now, some fields it'll be zero, other fields it might be a lot more. But it's important to remember, both with cover crops and this soil health stuff, there's going to be a plateau where that is going to, stop working so well, and we think that might be, 8 to 10 years or so. And as we increase organic matter in these soils and increase the natural level of nutrients in the soils, if farmers don't reduce the amounts that they're applying, the soil health has the potential to increase nitrate loads.
Great, thank you. Another one came in and a couple others. So any insights on changes in discharge flow rates over time? John asks.
They're increasing. Some of our streams have, since over the last century, the amount of volume that they discharge is almost tripled. The Raccoon is in that category. And so loss of perennial cover, the drainage, and the increased precipitation has greatly increased the discharge of our streams.
And Margaret, I'll try to summarize this into one question. So if I get it wrong, let me know. But Margaret, I think, generally wants to know what role has political policies played on why Iowa is so different in the Midwest from other Midwest states?
Well, we know that, policy has greased the skids for the hog industry to come in. And so North Carolina had a moratorium on hog confinements in the early 2000s, and that's when our hog industry started to expand. And so, they know this is a place where they're going to be welcome and that they can expand. And so policy definitely does have an effect. We also have policies that keep the price of nitrogen fertilizer low. you know, we built that big plant down at Weaver, Iowa, southeast Iowa, a few years ago. Those Egyptian guys came in and built that plant. We gave them big tax breaks to come and build that plant and provide commercial fertilizer for Iowa farmers. And so there's lots of policy things that we do that incentivize farmers to do what they do.
Thank you. And the last one I have right now is from Anthony, wanting to know essentially how do MMP and CAFO and other regulations, how does that dictate manure application and similar?
So the manure, when they create the manure management plan, they are allowed to use the yield goal formula for nitrogen application. And the yield goal strategy was discredited 25 years ago, if not more. And so that says if they can demonstrate a certain yield, like 200 bushels per acre, then they can multiply that by 1.2 for their N rate, which would be 240 pounds per acre. And we know that corn in Iowa after soybeans needs about 145. And so we also, if they're farming flat ground, the soil phosphorus does not limit how much manure they can apply. They're allowed to apply at the N rate. And so consequently, we get manure nutrients applied at far more than the agronomic rates here in Iowa.
And I think our last question is from Ken. Do you think promotion of CRP seedings on floodplain soil is beneficial or can be beneficial?
Yes. And so the one thing we know works without beyond a shadow of doubt is retirement of land. And so retire land. If the public wants to spend money, spend it doing that. And if anybody wants to ask, if you want to move on, they can put it in the chat and I'll answer it while the other speakers are going.
Right. Thank you, Chris. We're about a minute or two ahead of schedule, so that's great. So it'll give me a chance to mention to folks that up next we have a few speakers in a row that have been funded, at least in part, by donations to RRWA or into our research fund. And so one of these is Paul at Central College. He is a biology and environmental science professor. He is going to talk to us today about some work that was done partially last year and other work that he's done in previous years on monitoring cattail species, and then looking at like how conditions have changed at the wetlands, and then like establishment of invasive species. And so, Paul, whenever you are ready, the floor is yours.
Great, thanks a lot, Ben. All right, let's see if I can't get my technology going here. So I'm going to share my screen. Okay. So as Ben mentioned, I'm a faculty member here at Central College in Pella, and at a small college, I wear a number of different hats. And so I teach things like our general ecology class, our introductory environmental science class, but I also get to teach a couple of specialty classes, things like ethnobotany, which deals with plants, a field botany class, and a limnology, which is aquatic ecology. And in those, I get a chance to know the students, and we'll sometimes do some field research, and both those teaching activities and also a couple of other ongoing projects kind of led me to where we are today. And so my talk is about monitoring cattail species. And part of the background of that is that in the past, I did some work looking around the state for another invasive species called purple loosestrife. And I did some follow-up to monitoring efforts that the Iowa DNR had been doing around the time of the turn of the millennium. So I revisited that about 20 years later, and I went around the state looking for loosestrife, and I presented that at the Iowa Academy of Science. But also, I've also had a blogging project, which is a public outreach about Iowa's wetlands. And the gimmick is that I go to every one of Iowa's 99 counties and I profile a wetland in each one. So I've done that project, and that's a blog mentioned in my bio. And so I'm happy to talk about any of those projects. If people have questions, I'll leave some time at the end here for questions. But the idea is that as I was doing these various other activities around Iowa, and also some of the papers I've been reading lately in the literature about wetland ecology suggest that there's a change that's been going on basically in the last 20 years or so, where the cattails that we have growing in the Great Lakes, upper Midwest area are changing. And because cattail is such a widespread and familiar species, everybody knows cattail, it tends to go underneath the radar and maybe hasn't been as well recognized. But the project here is to do some monitoring in our local wetlands. And I did this work with Anna Shapiro. I'd like to thank the Raccoon River Watershed Association for funding to help me and Anna to do this work. It's preliminary. We did just a little bit of work last summer, and I'll be talking about that. But Anna has since graduated from Central College, and now she's actually moved on to a master's program at the University of Northern Iowa. And she's working with a colleague that I have there who also studies cattails. And so she's kind of carrying on and ramping up that work in other ways that are pretty interesting. So wetlands get a little bit of buzz. And so basically as background, wetlands are an interesting ecological transition zone where we have some aquatic and some terrestrial characteristics. They're not really water and they're not really land. And there's a long technical definition there, but basically wetlands are places where you get wetland plants growing, more or less. And it intersects with a lot of things like we were just hearing about with nutrients. Wetlands are a big player in understanding nutrient dynamics in our waters. Wetlands are big in climate change. But today I'm talking a little bit about the plants growing there and the effects of plants on ecology of wetlands. So our native species of wetland, cattails rather, are the Typha latifolia, the common or broad-leaved cattail. And I'll show you some photos in a minute of these. But we also have growing here at least a few individuals of a species associated with the coastal areas of North America, of the narrow-leaved cattail or Typha angustifolia. And it's perhaps not native to this continent at all, but certainly not native to the Midwest. But it has moved into the interior of the continent. And in doing so, it has actually been hybridizing with our native cattails. And the hybrid is recognized the name as Typha glauca. And there are different common names for all of these things. But the real question at hand is when you go out into a wetland here in Iowa, which of these three cattails are you likely to find? And you're going to find cattails in most Iowa wetlands. And so right here in the Raccoon River watershed, there's a site, not where we did our research for this project, but I had visited Finn Pond, which is in Greene County. And you can see there, basically, it's a sea of cattails and you know, acre after acre of that shallow water and mud flat covered with the spread of these cattails. So that's what we're looking at there. That's a pretty common site. So the cattails appear to be changing, and the biology is such that these different species or their hybrid have different characteristics, and I'll show you a couple of photos in a minute. But what you're looking at me holding there in that photo is the female flowering spike. And so that's got the fluff that has all of the little seeds in it. And one of those spikes can produce many, many seeds, of course. Above the female spike, the brown spike, you can see that there's a bit of stem that is naked, but it had the male flowers on it packed together in their own spike, and that was producing the pollen. And so one of the ways to tell which of the cattails you're looking at is to look at how wide those reproductive heads are, and also to look at the gap in between the male and female flowering spikes. And in that photo you see there that I'm holding, there's no gap. And so basically that has at least the overall appearance of a broadleaf cattail. And of course the leaves are pretty wide as well. But all of those characteristics that you see in those in the field, and we can take precise measurements with a caliper and so forth, all of those are highly variable from one plant to another. And it can be difficult to put your finger on exactly taxonomically, technically, which one you're looking at. So we did those field measurements, but many of my colleagues have turned to looking at genetic fingerprinting, molecular biology techniques to try to parse out which cattails you're looking at in a particular spot. But regardless of which one you're looking at, their biology is essentially the same. If you've got mud, a cattail seed will find it because the fluff with those seeds attached blows everywhere. People plant cattails, but I don't know why. They will find your wetland. They will find your ditch. They will find your pond. They will find them. And they're very adaptive. When they find a spot, they will grow with what's known as modular growth. And so there are these horizontal stems just underneath the soil surface that spread through the mud. And then the tillers, which are the green leaves, come up in great numbers. And so they can spread through an area very, very rapidly and can tend to dominate and pushing out other plants. And so reducing the overall diversity of plants in a wetland. And the question is also, is that going to change how the wetland actually functions? Is that going to matter in terms of the chemicals cycling, the use by different insects, bird use, mammals, fish in the water, and so forth? So those are all questions to be thinking about as consequences. Okay, so I'll talk more about these sites, but let me go ahead and show you. I did call up a photo. There's a great website with photos and information about Iowa plants. And so the plants here pictured show you some of the characteristics between the different species of cattail. Great photos, and there's photo micrographs that show pollen and everything else. So this is a wonderful website, and you can look at the Iowa Plants website and you can get these wonderful photos. Recommend that.
Hey, Paul.
Yes.
We're still looking at your PowerPoint screen.
Oh, I'm sorry. So I have to screen share the right thing. Got it.
That's fine.
All right, let's see if I can do that. All right, so this is the photos. If you've seen cattails in the field, what you're likely to look at is the female seed-bearing or pistillate spike, which you see there in that photo. But there are these other characteristics. There's a great photo that shows the gap between the male and the female spike there. Whereas on the right, we have the native cattail where there's no gap at all. So those are some of those characteristics. But this site has great photos. IowaPlants.com, recommend that. So Okay, we're back to the PowerPoint.
Yes.
So, thanks. So the three sites that we visited for this work, Christie Pond in Carroll County, McCord Pond Wildlife Management Area in Guthrie County, and Voas Area in Dallas County. And looking at those questions about what we found. And the answer is, sure enough, we're finding that cattails are growing in great numbers in those sites. Here at Christie Pond, we did have about 50% of each. So we had about 50% of the broadleaf cattail and 50% of the hybrid cattail, and is often found in other sites and in other studies, the deeper water tends to favor the hybrid cattail, and our native cattail tends to be found in the shallower water. We did find numerous birds and amphibians. I did make a report for the Watershed Association for those who read that, but we found burrweed, smartweed, nutsedge, rice cutgrass, river bulrush, and other plants, and then the mud flat with a little bit of open water. It had been dry there that summer. At McCord, there was ongoing construction, looks like a channel and an area for boats to put in, but we did find in the large basin there that there was hybrid cattail exclusively. There's a small basin that we found there that was more diverse. It did have a few cattails, but it also had many arrowheads, burrweed, carex sedges, and so on. We did do a few of our standardized data analysis on plots there at that site as well. And the plants did track very well with other work that we've done previously. And then the site in Dallas County had two large basins that we looked at up at the far north end of the property. And of those, there's a smaller at the very far north end that had more diversity. And we found a number of plants growing there, including cattails, but others as well. But then the larger basin, which was deeper, was dominated with great numbers, great spread of the hybrid cattail. At all of these sites, we did make some observations of birds, and we heard amphibians and so forth. And so these are healthy wetlands. And so one of the great questions I'm interested in is, what are the ecological consequences of moving from one of these cattails to another? So we're hopefully going to be continuing that work, my students and I, in the future. When I think about these cattails, I think about them as being basically a structure for other species. Muskrats would be the most famous, but when you think about organisms like damselflies and dragonflies, they're going to be using these as well. There's going to be all sorts of birds that nest in them and so forth. And so I'd like to learn more about those. I did say I'd leave some time, so if you have any questions, I'd be happy to answer those.
All right. Thanks, Paul. So there is one, at least I have one, and so there's a few others are popping in. But how long does it take for these hybrid species to become so widespread? Like, is it like over a few years? Is it really quick? Like, how quickly does this happen?
So my observations, just anecdotally, are that the battle is done and the hybrid cattail has won. You can certainly still see the native cattail, but if you go to any typical Iowa wetland at this point, it's going to be at least half, if not dominated by the hybrid. It's actually fairly rare now to see a good stand of our native cattail. And that happened, I don't know, I assume that happened over the course of about 20 years. I don't have good data from before I moved to Iowa, but it seems like it happened relatively quickly.
That's really quick. Wow. So just for people to be aware, We are in our break right now, but if you have questions as you have questions, please continue to send them in, and we will use this the 4 minutes also if you have a break.
And I'll follow up with anyone individually as well. So I'll check the chat here in a second as well.
All right. So another question is, you mentioned somebody wants to know about the impact on wetland diversity overall. As you're going to now the more of the hybrids, like, is that having an impact on what animals are living in that wetland?
So that's one of the questions I do want to study that I'm hoping to work on. Again, sort of anecdotally, what I'm seeing is, at least for many of the common birds that you associate nesting with, I'm not seeing a particular impact. So wrens or blackbirds that tend to nest in these, I'm finding them working these hybrid cattail stands just like they would have been nesting in the broadleaf cattail. I haven't seen any suggestion that they're affecting the insects, but others, and I think I also have seen that it is affecting probably the mammals. So particularly things like muskrats probably do favor our native cattails. Others have suggested that, and I think I'm finding that as well.
And then a question from Matt wants to know that with the proposal from the Nature Conservancy, will you be involved with wetlands along the Cedar River?
I'm not familiar with that. I'll have to look into it.
Yeah, I'm not familiar with that either, so that's interesting. So you said also that you run a blog. Was that link anywhere, or can you put that in the chat? The 99 wetlands.
Yeah.
But those that I follow it periodically. But another question or a follow up. There was a proposal from the Nature Conservancy and the NRCS to establish wetlands in 2-year floodplain along the Cedar River.
Patrick, Andrew, if you could put that link in the chat, it might be interesting for all of us. Thanks, Paul, for your link there.
Yes, thank you.
And I'll add the blog as well.
That's great. That's great. For those of you at home, you might want to stand up, stretch, turn around, and sit down again. We're getting close to our 10:30 time. The chat is available for that networking that you've been doing, and any other further questions for Chris Jones and Paul at this time. We're happy to have 50 people on, and that you would join us on this in my part of Iowa. Beautiful sunny morning, little chillier than I expected. And I don't know how it is at your place. I'm going to do that turn around, fill my cup of coffee, and we'll start here in just a second, right, Ben?
Yeah. But thank you for that, Paul. I personally found it interesting. So Storm Lake has made a constructed wetland, and it's been fun to watch it go from just being established with essentially nothing to now it's artificially diverse, but it has really taken hold over the last 4 years that it's been here. So it's been really amazing to watch it transition from nothing to what it is now.
Yeah, constructed wetlands are a whole nother can of worms. But one of the challenges I think we face is that when we engineer sites that are going to meet the requirements to count as a constructed wetland, we want to make darn sure that we've got plenty of water all the time. And so we tend to engineer it so that it has basically great conditions for cattail takeover, deep water all the time. So I think that's part of what we're fighting. And also, there's a cause and effect question here about things like sedimentation and eutrophication with all the nutrient runoff, and how that might be favoring cattails because they're really good at taking up all of that, more so maybe than our native species. So what's the problem and what's the symptom of the problem, I guess, is a question. But
yeah, it's interesting. It's so again, thank you. I appreciate that. And again, Paul was the first of our lineup that has been funded in part by Raccoon River money and donations. The second is Peter. And I guess apologies for having you guys back to back. There's been a few biblical jokes in that when you're saying this up, but Peter Levy is with at Drake University at the teaches environmental science, and he will be talking to us about some of his work that he's been doing with some of his students.
Great, thank you, Ben. Chris and Ben, since I can see you, do you hear me and see my red pointer on the screen? Fantastic. Yes. Thanks for the invitation. It's always a pleasure to chat with the Raccoon River Watershed Association and associated friends. I really do appreciate everything the RRWA has done in the watershed, and it's such a great testament to your organization that you fund research because you want to know what's going on in the watershed, and you really support a lot of undergraduate science, which is something that we do at Drake, of course. So thank you for your support, and it's a pleasure for me to chat with you today. Also, happy vernal equinox, everyone. I hope that you're enjoying the change in our calendar. So I wanted to talk today about some research, not funded through RRWA. We have some of that going on now too, but rather some exciting things that are going on. And I knew with Chris speaking earlier today that this would fit in well with that. The title here, Heterogeneity in a Homogeneous Watershed. I'm going to explain that in my first slide next. But what I want to describe today is our new project where we're exploring water quality by the meter and minute using this aquatic drone that you can see in this image. And I want to acknowledge my co-authors and colleagues on this project. These 4 scientists are at Oak Ridge National Lab in Oak Ridge, Tennessee, where a lot of the engineering and ideas for this aquatic drone were born. In particular, I want to acknowledge Natalie Griffiths, who is second from the left here, who has done a lot of the work. She's an aquatic ecologist there at Oak Ridge. So it's exciting for me at a small private liberal arts school where our emphasis is on teaching to be involved in a pretty major more sort of R1 research opportunity here in central Iowa. So my title was Heterogeneity in a Homogeneous Watershed, and I really enjoy starting talks with this NASA image. This was taken in May of 2012, and in it you can see how homogeneous much of the Corn Belt is, especially before the corn starts growing, the corn and soybeans start growing. So you see southern Minnesota, much of Iowa, much of Illinois, eastern Nebraska as very brown and very much in the bare soil category. And so we have this very homogeneous watershed. We saw some amazing data from Chris, where there's a lot of nitrate exported from each one of these sub-watersheds in Iowa. And so we know that this problem is consistent over large spatial scales across the entire state. To put it into a little context, this is a figure from a book on limnology, freshwater ecology. And here the authors describe how different parts of a stream or watershed develop or change both by time along the x-axis and shows the spatial extent of these different parts of a stream or river along the y-axis. And so what you can see here is that at a watershed scale, when we're thinking about the Raccoon River watershed, of course, over natural geologic time and over natural geologic processes, we'd expect the watershed to develop on the scale of thousands, if not tens of thousands of years. However, with the human impact on these watersheds, we are accelerating the change in the watershed to be on the scale of decades or at most 1 or 2 centuries, since humans have really modified this into industrial agriculture. Now, as we consider these major impacts, we want to start improving it. And there's a lot of stream restoration work. This stream restoration work often operates at a reach scale. So when we think about a stream reach, we're talking about tens of meters, maybe 200 to 300 meters, but not nearly as much of as big as a stream section, or not nearly as big as an entire watershed, of course. And we're trying to do these restoration efforts in a very accelerated manner. So oftentimes these are put out onto the landscape in less than a year. And then we hope that they somehow improve water quality or meet those objectives that are spelled out in the funding proposal that leads to those restoration efforts. What I wanted to talk about today is one such major restoration effort in Alleman Creek, Iowa. So this isn't quite in the Raccoon River watershed. Rather, this is in the 4 Mile Creek watershed, which then drains directly into the Des Moines River just south of the city of Des Moines. And in this small headwater agricultural stream, we're studying a 2.5 kilometer stretch here, so about 1.5 miles. And right now in this 1.5 miles of stream, you can see that there's 26 tile inlets along that stretch. And you can see that it's pretty heavy agriculture. By the end of this year, if all things continue to go right, there will only be 2 tile drains left in this 1.5 miles of stream. And so this is a major restoration effort on a very small scale that's occurring, largely in part due to some funding that Polk County was able to get, as well as some fantastic landowner support for this restoration effort. So here you can see a tile drain coming out into Alleman Creek. By the end of 2021, and hopefully even sometime this summer, 24 of these 26 tile inlets will be converted from a traditional tile outlet, as you can see in the left side of this figure, to a saturated buffer, which you can see on the right side of this figure. So the idea behind the saturated buffer is that the tile water is intercepted before reaching the stream directly, and rather that water is diverted along the buffer, and then there's a much longer flow time and residence time for the water as it moves through the saturated buffer towards the stream, rather than the very quick route through the pipe. As the water takes this much more sinuous and longer path from the field to the stream, bacteria are able to remove some of the nutrients, such that the water entering the stream then should have less nitrate than if it entered directly through the tile drain. So that's the conservation and restoration goal with saturated buffers. When we think about how we can measure responses of streams or rivers to water quality, there's a couple different techniques that have been done. I did want to just put this slide in here to say that our goal by this time in 2021 was that we'd be able to compare these different techniques more robustly. But as we all know, 2020 was a strange year in many ways. And one of those was that we had quite a drought here in central Iowa, especially in western Iowa. Alleman Creek, as you see in the image I just animated in, did go dry starting in later August, and it remained dry through January until it was thankfully covered in snow. So that our idea entering this study in 2020 was to be able to compare these different techniques, though we have some limited data from 2020, though we're already starting here in 2021 to get some good data. So these 3 different techniques that I just wanted to briefly show you this schematic on is that we can think of sampling streams and rivers in a traditional way, where you could send a team of scientists out to collect multiple samples within a watershed. That's what these red dots indicate. And then those samples are transported back to an analytical laboratory. And at that analytical laboratory, we could find out the concentrations of nutrients and such. Because there's a fair amount of people hours involved here, this isn't something you could do every day, but you could set up a schedule where you do it weekly or bi-weekly, and you get a pretty good snapshot of the watershed. Another way that we can measure water quality within a watershed is, as Chris so nicely described and demonstrated earlier today, is that we can deploy sensors. What's great about sensors is that we get data continuously. So these blue bars here on the bottom are a representation of frequency. And so oftentimes we can set these loggers to log every 10 minutes, every 15 minutes. But the issue here is that they are generally costly. And so we might only be able to measure one point in a watershed. I will note that when Chris showed that map of Iowa, we have one of the most monitored states in the country. We are one of the most monitored states, which is really fantastic. Still, with only 72, there's still some gaps, especially at those smaller headwater streams where we don't know quite as much about the water quality without doing something where we might send people out to grab samples. So then the third way that I wanted to present to you today is sort of a combination of the two, using an aquatic drone to get a much greater spatial snapshot. So be able to monitor multiple streams within a watershed with high frequency data rather than just one point. But of course, because there is these are not autonomous, so there is some people hours involved. So you can see that the blue frequency bars here on the bottom this isn't something that could be done every 15 minutes, but it is something that could be done weekly or every other week. So what I want to do today is to compare some data between these sampling techniques, focusing mostly on the data we've been able to collect with the drone. So here's an image of the drone, which we've aptly named the AquaBot. And what we're able to do with this is quantify a lot of different water quality parameters at minute and meter scales. So here we have a multi-probe sonde that's collecting data for dissolved oxygen, pH, conductivity, and turbidity every minute. And then on the right side here, we have a real-time nitrate sensor that's collecting data every minute as well. All of these data are piped into a data logger. There's a GPS unit that's providing pinpoint accurate GPS data at the minute scale. And then we're also collecting light data. This isn't very large. You can see the specs here. It takes about 2 people to carry, but it's just a couple feet long, couple feet wide. And all of these sensors are wired to a data logger that has Wi-Fi. And so here are some screenshots of my iPhone as I'm walking along the stream bank next to the AquaBot. So we're able to see all of these parameters changing in real time on our phone. What's really great about that then is as we notice changes within the stream water quality, we can spin the drone back. We could keep it in one place for a few minutes. Here you can see we nosed it up to a tile outlet. Right now we are operating this manually, but autonomous operation is possible. That would, of course, could only happen maybe at a reservoir or a larger river where you could have it steer itself. But currently we have it set, so we do need people out there operating it. And the measurement frequency is one set of measurements every minute. But as you can see, one set of measurements is a lot of different data points. Here are just 2 quick videos showing the AquaBot in action. This is in Walnut Creek, which is in the Raccoon River watershed. We were just testing it out. That's the AquaBot going full bore down the stream. Of course, if we're going at high speeds and it's only collecting data once a minute, we're getting data about once every 40 or 50 meters, which isn't ideal for seeing that really fine scale variability. Here you can see we're nosing up the AquaBot to a tile outlet. And so we'd be able to see how does that water quality vary right there at the tile outlet. And so you can see it operates pretty smoothly once we've had some practice with the remote controls. So if we were to contrast some of the different ways that we can collect data, I'll say that I had an idea that Chris was going to show you how fantastic sensor data can be. Of course, that, as I mentioned, sensor data is often good at broad spatial scales. And with this amazing network that we have in Iowa with 72 different sensors, we're able to look at all these different sub-watersheds across the state. If we were to take a look at some manual sampling data, sort of that first option that I described, where you could send a field team out to do grab samples occasionally throughout the year, we've been doing that at Alleman as well. We've been sampling upstream at a midpoint in the watershed and then downstream of this 2.4 kilometer stretch. And here's some nitrate data from those 3 points. Blue is upstream, yellow is in the middle, and the red is downstream. What you can see there is that the advantage to doing this sort of water quality monitoring is that it's the lowest effort. With nitrate, we can see a really clear temporal pattern that nitrate is often high in June, and then into July and August it crashes, stays low for those fall and winter months before it starts to rise again later in the fall, early winter. And then the other advantage of this is that we can continue sampling during low flow periods. So there's a big period here where you can see we only were able to get water at the upstream site, and that's because the rest of Alaman was dry. We cannot operate the aquabot in less than about 12, 15 inches of water. So we don't need a lot of water to operate the aquabot, but we do need some. If I zoom in here, though, we can see one of the potential disadvantages to just doing grab samples, depending on how well you want to know this stretch of stream. You can see that there's variability among the sites. In general, the most nitrate in terms of concentration was downstream. But here you can see this date in June, the sites were flipped. So the lowest nitrate concentration was at the upstream site, at the downstream site, and the highest was at the upstream site. So we see that there's variability among sites, and then also the concentrations are often inconsistent. So here we see on this date a big variation in concentration across the 3 sites, whereas on other days we can see that it's a relatively tight window of how variable those concentrations are. So what are some advantages to the aquabot? And now some data on those. I wanted to start with a very expected figure here. So this is stream water temperature on the 3rd of June. We start here at the upstream point and we run the aquabot downstream for the 2.4 kilometers. And you can see that stream water temperature increased, right? From when we started in the morning, it was 14 degrees. And by the time we pulled out at the bottom in the afternoon, it was almost 20 degrees Celsius. So of course, there are changes that happen during the day that we expect. I will say, because I'm expected to get a question on it, how do we account for that variability? We do have 2 sets of stationary sensors now that we just deployed and got online for the 2021 field season. And so now we will be able to compare our high frequency data along the reach with these 2 stationary sensors, both at the upstream and downstream points of this reach. So we will be able to account for changes in temperature and flow across the period that we're running the aquabot. Here is specific conductivity. So in general, specific conductivity, which is a measure of the ion concentration within the water, in general, that decreases. You can see the purples and blues at the top part of the reach, and then more of the yellows and oranges downstream. If we were to look across the 2.4 kilometer reach and look at concentration, so here's the top of the reach at 0, and then distance downstream down to 2.4 kilometers. All of these dashed lines represent some of those 26 tile inlets. So we have 26 dashed lines here. You can see that in some locations, the conductivity is highly variable around those tile inlets. And some different patterns that we noted is that high variability where 2 tile inlets come in at this road crossing. And otherwise, in general, we see a decrease in conductivity as we go downstream. You can see that along this stretch of the stream here. And then in this more sinuous stretch, this is property, Lee Tesdell's property. He was so kind to let us work on his property, and he's joining us today. You can see the stream is more sinuous across his property, and you can see the cover crops there as well. There's a little bump just at the beginning of that property, but then you can again see that characteristic decrease in conductivity downstream. If we look at nitrate, we can say that there's nitrate spikes throughout the reach. Here at that road crossing again, here at another tile inlet. But then what's interesting with these data is we can see that there's some spikes not associated with tiles. So this could be a good point of reference to think about where should we put management effort if there's some nitrate spiking without tile inlets. I see that I'm close to my time already. Is that what you were going to tell me, Ben?
Yes, sorry. Go ahead.
So I'll just wrap up here. I'll say that when we do multiple aquabot runs, here you can see aquabot runs 3 weeks apart. The concentration was generally the same. Here are 2 points that were both highly variable, but they varied between the dates, whereas other sites like this black circle, the concentrations were more consistent. Finally, we walked this reach and grabbed samples every 100 meters. You can see that there's far less variability when we're just grabbing a sample every 100 meters, and it took the same amount of time, in fact, more time to walk without the Aquabot than it did with the Aquabot. So what we're doing now in 2021 is we're continuing weekly and monthly Aquabot runs. It'll be especially great to evaluate the efficacy of those saturated buffers. We'll then also be comparing that Aquabot data to these stationary sensors and that manual grab sampling method. And then finally, we'll be examining spatial patterning along stream and river reaches throughout the network as we run the Aquabot on parts of 4 Mile Creek as well as the Des Moines River. So I know I'm out of time. I apologize for that. But I'm happy to take questions. There's my email in the upper right, and I'm happy to respond even next week or whatnot, or in the chat now.
All right. Thank you, Peter. We are now just a little bit behind, so we'll move on to Adam. Peter, if you would be kind enough at the end to stick around, if that way, if there are questions that people have for you or other speakers, we can do it that way. Is that okay?
Absolutely. Yep. I'll be
all
here.
right. Thank you. Chris, you're up.
I am. Thank you. And thank you, Peter. I want to assure all of our participants that we do monthly meetings that are open to the public and usually have a great speaker. And I think Peter is a good example why sometimes we need a little bit more time than the time that we were able to figure everybody would sit still for this morning. Hope to see you back soon, Peter. Thank you so much. I'm here to introduce Adam Dixon, who has a fascinating project that I was lucky enough to participate in by having little recorders in 2 of my farm fields. And this will be the first time I'll see the results. And so take it away, Adam Dixon.
Thanks, Chris. Can everybody hear me okay? Okay, great, thank you. Okay, so yeah, so I'll describe some of my PhD research. I recently defended this work in December, and so I'll officially graduate this May. So I'll present 2 of my chapters today. Hopefully, I hope it's not too rushed, but I will be speaking sort of fast, so I sort of apologize in advance. So I would just like to say that, you know, the reason that I got here and that I'm presenting this research in the first place is because where I'm actually from, which is not Maryland, it's Kansas, in the Arkansas River watershed, is because I really like to fish and swim and hunt as a kid. And we have EPA regulations on fishing and swimming in the Arkansas River. And so when I was thinking about career choices, I was pretty distraught by the fact that I couldn't swim or fish in my local river, and it sort of led me on the path that I got to today. So I ended up at the University of Maryland, which is Baltimore County, which is like quite a ways away, but I was able to sort of channel that work back in Iowa because I wanted to investigate social and ecological relationships in intensive agricultural landscapes, and my PhD advisor supported that. So I was really lucky to sort of do this research, which is what I tried to design it as representative of Corn Belt agriculture. And so that's sort of represented by this map where the brown light shading is crops in the US, cultivated land, and then Iowa, obviously, right in the center of the Corn Belt. So a perfect place to study interrelationships between wildlife and intensive agricultural management. And so my dissertation research in sort of general was trying to sort of create this framework where I'm measuring landscapes, measuring habitat, measuring wildlife, and I'm also measuring social process. And that's sort of another chunk that I won't describe today, but it was one of my chapters, and it's sort of why I set this up the way I did. So what I really focused on in the work was the green parts of the grid. So the grid on the slide is like basically a map, right? And the green is like the non-crop vegetation area, and the yellow is corn, and then the orange is soybeans, and then you have a few other land uses. And so I wanted to focus on the green part, the non-crop vegetation. And so I used satellites and drones and acoustic recorders, which are all kind of like novel technologies. And so I wanted to investigate methods of how you would actually set up a research framework like that in an agricultural landscape, and then get to the actual sort of effects of management and the effects of the observations of what I'm seeing. So so I measured non-crop vegetation area and configuration, which I'll explain a little bit, and also tried to measure agricultural practices and put those into sort of a statistical framework. So I got started on this research during the summer of 2018, where I had 12 observations, so a very small number of observations, but I was just getting going. I didn't really know what I was doing. But I came to Iowa. I met some folks at the Iowa State Research and Demonstration Farms, and they allowed me to set up my research there. I reached out to TNC, the local TNC chapter, and I was able to get a recorder on a TNC site in Northwest Iowa. BKG is in the map. You can see that's Broken Kettle Grasslands. So I set up my recorders in these landscapes, and you'll see the black circles around the red dots where the recorders were. The black circles are basically my landscape, what I'm actually measuring. And the reason I chose that was because that was the area that the soundscape and the landscape overlaps. And so if I'm listening to birds and I'm trying to decide what the scale of my study is, I set it up to be relational, like perfect, like, you know, one-to-one. So the soundscape and the landscape are the same. And so I'm going to look for statistical relationships there. So I wanted to, in this first project, I wanted to measure composition and configuration. And so landscape composition are like the different crop types, the different vegetation types, and configuration are the shapes of those sort of in the map. Are they square? Are they rectangular? Are they big, small? Are they isolated? Are they linear? So a lot of vegetation patches in the landscapes I was investigating are, you know, field margins. And so they're very linear. And so I wanted to ask if that made a difference. And so as I mentioned, I used drones. I used a new satellite system. It's a microsatellite. So on the lower left-hand corner, it's like 10 centimeters by 30 centimeters. And there's 150 of these. I think almost 200 now. They keep launching new ones up there, but they're continuously monitoring the Earth at pretty high resolution, but not like UAVs or you know, drones. And so what I was wanting to ask was, does that make a difference? Does the resolution at which I can map the landscapes make a difference? And you can see on the right side, I'm actually picking up weeds and stuff in the fields and the field margins, whereas on the lower one, it identifies the crop and the big vegetation patches very well, but I don't get that fine scale measure. So the question is, does that make a difference? And so then, in terms of listening to the birds, I wanted to generate a measure of the bird population. And so the most simple measure is species richness. When you're listening to birds, you don't have that visual component. So I had to sort of redefine what species richness was. So I called it vocalizing bird richness, because those are the birds that I was able to, those were the species that I was able to listen to. And so I tried a couple different ways to listen to what I was hearing. I obviously can't listen to everything that I hear. And so I used what are called an acoustic index, which takes the sound file and it converts it into a unitless measurement of how much activity is going on. And so I tried out a couple of different ones. I settled on this one called the Bioacoustic Index, which was developed some time ago because it kept out rain and wind very well. And I listened to, so I took those values that I got from that, and I listened to the recordings, only the recordings that had really high values. So I listened to 100 of them. And then, so that was one approach. And then the second approach was just to listen to birds in the morning, which that's the traditional approach. Ornithologists go out in the field and they go in the mornings, you know, like they go at 5 a.m. just right at dawn. And so I listened from like 10 minutes before dawn to 2 hours after, and counted how many different species I heard at the point. And I just like to point out, so the RRWA funding went to purchase the recorders. So it was really great that I was able to increase my sample size significantly based on the contribution. So I'll say thanks a couple times, but it was a huge contribution. It really helped me get my research going. But I will note that that was for the second year. I'm getting kind of ahead of myself here. But these are my results, some of my general results. I apologize, I'm going to move this pretty fast. But what you can see here is that first, I'd like to point out the configurational measurement, which I have as circle in the circle column. I found that the measures of configuration that were invalid because the landscape was so homogeneous that I was getting sort of erratic responses. As you can see in the circles, it's a completely different relationship between the drone, the unmanned aerial system, and the satellite measurement. And so I was concerned by that. That was obviously a big concern. But I found that the measure of area, non-crop area, was statistically significant and made sense. And so that's what I decided to focus on in this first chapter. I realized that the measure of non-crop area is reliable and shows statistical significance. And so that's something to focus on. And so looking at just a regression, a simple univariate regression relationship, I found that the drones were explaining about 54% of the variation in the data. So pretty good. And then 16% lower were the satellite measured data. And so the question is, does that difference matter? And what I kind of decided is that it does matter, but if you're going to scale this kind of research up, drones are inefficient because it took a lot of time to do it. It was a little bit more expensive, and you would need more people. One researcher wouldn't be enough. So I'm sort of buzzing through this, so I apologize. But I found that the dawn sampling was better than the index approach, and so listening to birds in the morning is better than using an index to sort of try to evaluate all the recordings over time. And that composition, my area measurement, was a simple and effective measure, and that the configuration didn't add value here because the landscapes were so homogeneous. Also, obviously, that I just said that drones UAS results showed a stronger relationship than PlanetScope, but the PlanetScope, the satellite data was free and easier. And I detail all this so I was able to get this research published in the journal Land, and so that's out. It's been out since like last May, so about 10 months now. So I scaled the research up with the infusion of support from RRWA. The next summer, I was able to measure 60 landscapes to sort of bring this sort of vision of agricultural landscape and soundscape research to across Iowa. And I was only able to do it with collaborators. And so, I'll talk about more of that later. And so the way I set it up in this version this summer was that I learned from what I learned from the results the previous summer and tried to improve upon them. So I asked, can I improve the non-crop vegetation maps from the satellite data? We saw that the results were a little bit lower, but maybe that's because the mapping methods weren't as good. So I tried to improve my mapping methods. And I wanted to look more into the measures of non-crop composition. Is there more value to compositional measures, since configurational measures weren't that informative? And then the question is, can acoustic data collection be collaborative? Because if you're going to scale up this research, really, there's no reason why one person has to do it all. Like, it can be shared. And so I worked with, I sent 17 farmers, 28 of the recorders in the mail in these boxes. And then I reached out to other farmers and landowners to get permission. And then I worked with the research and demonstrations farms again and sort of set up my 60 research sites. And so in total, 24 people helped with the effort. And these are just some pictures of the sites. This is one of the, an improved prairie strip. This one is not, this is in the Missouri River floodplain, where there's obviously just a very minimal vegetation in the field margin. So I was trying to get the variety. These are some of my other landowners that helped set it up. I photobombed myself in the upper left-hand corner, so I thought that was kind of funny. And then with each recorder, I added a recorder survey, as like, what were the management practices around the recorder? And one of the most impactful measures of management in the literature is pesticide application. Beyond land use, pesticide application is thought to be the biggest contributor to bird population declines. And so I wanted to measure that. And so what I found in the end was that I got 43 successful recordings out of the 60. So that's about a 72% success rate. This species accumulation curve kind of shows, like, as the line flattens out, that tells me that I'm listening and hearing the same species over and over again. So if I listen to almost 100 recordings, that I'm pretty much sampling the bird community that I'm going to hear. And so that sort of told me that that was enough recordings to listen to. Okay. These are just some of the maps that I got. They were a little bit more accurate than the previous maps, but they can, you can really see the non-crop vegetation interspersed with the agricultural fields at my sites. And so I wanted to add value to the compositional measure. And so I found these images, this idea of image texture. A lot of landscape ecologists are looking to see if image texture is like a convenient and easy way to measure landscape heterogeneity, and asking if that's related to the species that they find. And so they've done this at very large scales in different places, but not using fine resolution data in agricultural landscapes. And so I wanted to sort of take that approach here. And I won't go into the statistics or the how it's calculated, but just the idea that images have texture, and texture in images can be quantified, and it's thought to represent plant species diversity. So I incorporated that into my statistics. And so in all, I had all of these different variables. I had the non-crop vegetation proportion, which I found was significant during the previous summer. And then I had image texture measurements. And then I also created texture times area measurements. So I was basically creating, transforming the variables to provide a texture and an area measurement all in one value. I also measured pesticide application, which I mentioned, and then I wanted to include longitude and latitude as control variables. And so my results were that univariate models were the best performing models. When I included all the different variables, the model performance decreased. So I'm presenting here the 100-meter extent. That's where the landscape and the soundscape overlap. But I'm also presenting the 400 meter because I found that the relationship appeared to be a bit stronger at that spatial extent. So when I made the circle to a 400 meter radius, it appeared that that was kind of the area that defined the habitat better in this study. So I'm presenting those both here. Thanks, Ben. And yeah, so the results were a little less strong than before the previous summer. And so the question is why? I'll get to that in just a second. So the conclusions were that collaborations did decrease sampling success somewhat. It increased the complexity of the operation. The collaborators didn't know exactly what was going on. They were just really excited to help out, and I was excited to have their contribution. But it was a little bit complicated setting the recorders up, and moisture would get into the bags, and there's lots of different complications. So it did decrease the sampling success somewhat, but it was still, I think, worth it because I still got 43 good samples. When there were failures in the recorders, it was commonly because moisture got into the electronics, and so that made the recorder malfunction. And so to this question of why the explained deviance, why the statistics was less strong than chapter 2, and maybe it's because I just had a bigger sample size and that I had a better representation of farmland. It was also a slightly different time of year. It was in June. The June of 2019 was really wet. It was a weird year, perfect for my dissertation research. I lucked out, got an off year, but that's could have been a difference. But also, a lot of the recorders that also failed, another reason was because of wildlife. The wildlife would poke at the units, and so they were protected by a plastic bag, and that would allow moisture in. And so it was harder to measure the areas where there was probably more wildlife because the wildlife would tamper with the units. So it's kind of a funny thing, but it kind of screwed up my statistics. So, definitely a learning opportunity there. And so I found that image texture actually improved measures of landscape composition by just a little bit. So that was kind of interesting. And I also found that my simple measure of pesticide wasn't robustly significant. There was like hints that it was, but I think I need to measure it better than just as a one or as a pesticide was applied or pesticide wasn't applied. It really kind of matters what pesticide, how much, and when it was applied to, I think. So the future directions of this research are to bring sort of the framework full circle and look at the social relationships and how they interact with the wildlife relationships. But I have the ecological observations sort of charted out and how that would be done. And also have the social side as like how farmers view their role as a farmer and how that relates to the habitat and the wildlife that you're seeing. So that's kind of my overall goal of research. And so, so I'll sort of end there. My key takeaways are that keeping it simple works. The drones were really great, provided high resolution data, but the satellite data was more easily scaled, and that vegetation composition was more important than configuration. And then the last point was that farmer identity points to longer term benefits and agricultural conservation, but I didn't really talk about that much, but that was the other sort of contribution of my dissertation. So thanks a lot. This was really helpful to have the support. I'm sorry I went over time, and I'll end there. Happy to answer questions.
Thanks, Adam. We'll again save questions for at the end after Sylvia's talk. So Sylvia, you're up next, and the last of our talks for today. So again, just real quick, Paul, Peter, and Adam's research was all supported in part by donations and membership from you all. And Sylvia today will be talking to us about a brief history of Iowa's agriculture and conservation policies and practices. Sylvia is also at the University of Iowa in the Department of Geographical and Sustainability Sciences. So Sylvia? floor is yours, and take it away.
Thank you, Ben, and thank you everyone for being here this Saturday morning. I am delighted to be here with you. I would like to start by acknowledging the people who were here before and whose land we are now so intensively cultivating. What you see is the names that the Native American nations that used to live in Iowa gave themselves, not the names that colonizers gave them. These are largely taken from a book written by Lance Foster, who is the vice chairman of the Iowa tribe of Kansas and Nebraska. The locations of the tribes that you see were given to me by the Office of the State Archaeologist that I should thank. And what you see in the landscape behind is the 1850 General Land Office map. This is when Iowa was surveyed to be settled. So this is the vegetation that we had in Iowa pre-settlement. You see a lot of prairies. You can see the Des Moines lobe and the wetlands. And in 1720, Iowa was largely inhabited by people of the Oneota culture. So the Bahoje, we call them Iowa. The Jiwere, we call them Oto. And the Nudachi, we call them Missouri. There were many nations around Iowa that had hunting grounds. The Ho-Chunk, the Dakota, the Sanish, or as we call them, Arikara, what you see then, the Omaha and Ponca, and then the four groups are what we call Pawnee, but they never called themselves Pawnee. So in 1720, before displacement started, this is what the people who lived in Iowa, largely farming in the summer along the river valleys, and then hunting big game like buffalo in the winter, okay? And then as this displacement starts, lots of nations start leaving Iowa and coming through the Meskwaki and the Sac or Ozakiwook that you see here in the map were displaced from northeast of where we are now. The Dakotas are still hunting in Iowa at that point. When we get to 1840, which is where I'm going to start tracking things for you in a second, the Bakoja have left Iowa, and we start seeing Euro-American settlement. There is a war between the Dakota and the Sac and Fox, the Meskwaki and the Sauk and the Ho-Chunk are put in the neutral ground. So to me, because I am Italian, this is a very recent displacement of people who still have deep roots in Iowa. By 1900, there were less than 400 Meskwaki in the state. So this process that we're going to start discussing in a second was very swift and very brutal, not just to the landscape, but to the people who used to inhabit it. And this is the swiftness of the process. So what you see here on the right side, the brown line shows the improvement of land in Iowa. Even the words are loaded. The words are always loaded, right? By 1950 years, Iowa went from 2 million acres of improved or cultivated land that had been plowed, the forests that had been cut, to almost 30 million acres. And the other line shows that same process for all the states that you see in the large map bordered in blue. So again, we went from very low numbers of cultivated acres to almost 250 million acres of improved land. This process was bootstrapped by the land itself, and the state and the federal government used the land, sold the land through railroad land grants, school grants, Des Moines River grants, to create the infrastructure that would help settlers farm. So you see here the incredible fast growing network of railroads that we had built largely between the 1850s and 1900 to move a lot of the crops to Chicago for export. This model of Iowa agriculture has always been based on producing excess that has to be exported or sold outside the state. The changes in the landscape were not sustainable, not just from a social standpoint, but also from an environmental standpoint. So what you see here is details of the problems that this very aggressive process of land cultivation created across the state in terms of erosion and in terms of loss of habitat and so on and so forth, particularly in the driftless. So Dr. Maas will appreciate this slide. There were parts of the state, and Dr. Jones talked about this as well, where the karstic soil, for example, is such that we see immediate degradation of the landscape. And we've seen this immediate degradation from the beginning of white settlement in the state. So some of this degradation is what drove activities in the 1930s to start addressing the problems of our farming system. And this was coupled, of course, with the Great Depression and the Dust Bowl more generally in the United States. So we had our first farm bills in the 1930s. Mostly what I want to emphasize here is that from the very beginning, conservation is an ancillary goal to farm income support. And so what we see is these land retirement plans like the Agricultural Conservation Program. The goal of these plans was to reduce acreage, thereby propping up prices and improve environmental quality by taking marginal land out of production. But what I'm going to show you in a second is that second goal disappears when crop prices are high. So we have a yo-yo in our environmental policies in ag, because when the times are good, that these programs disappear and we go back to farming fence row to fence row like we did in the 1970s. So the ACP in the 1930s actually failed because farmers are not stupid. In fact, farmers are very, very savvy utilizers of federal and state agricultural and conservation policies. And so what happened was farmers just enrolled land that was really bad for crop production. They used the money from the conservation programs to more intensely grow crops in the good land. And so we actually never stopped having a surplus. This obsession with producing more is really driving a lot of the problems we have in the state. Instead of producing better, we just want to produce more, okay? So we had these first programs in the 1930s. They were largely forgotten in the 1940s because we were exporting to Europe through the Marshall Plan because of World War II. And then they show up again in the 1950s with the soil bank, because again, our export markets were dropping and we didn't know where to sell anymore. So we can also see that this system is propped up by subsidies. This chart from the USDA ERS shows in real $1 million, and we also have Iowa and Illinois, by the way. Iowa beats Illinois in the amount of subsidies. We're first in the nation on that too. We have high subsidies when crop prices are low, and then the subsidies go down. The subsidies go back up in the 60s when crop prices are again low and there's no export demand. Subsidies go back down in the 70s when we start exporting to Russia and we have a fence row to fence row, and then they go back up during the farm crisis. Now, what you see though, is that unfortunately, even in the years of the ethanol boom, look at 2011, 2013, the subsidies have stayed high. We now have subsidies baked in, and it looks like they just go up from a pretty high benchmark when the lean times come. So this is a big problem from a social welfare perspective, but it also tells us that society is subsidizing this system at pretty heavy levels. And so we should have a say on how that public money is spent. And we should get some bang for our buck. And we should also stop this yo-yo nature of conservation programs. So the land that was all improved, you can see here in the incredible rapidity of that improvement of the land in the first in the last 50 years of the 19th century is the land that we still grow crops on right now. So you can see the importance of corn from the very beginning, how wheat declines rapidly in Iowa, and we have the introduction of the corn soybean rotation really becoming the norm in the 60s and 70s. This landscape has seen a lot of changes, but it's a very heavily used landscape. First, it was used with horses and mules, and then as the magnificent work of our land grant institution, that happens to be my alma mater as well, paid its fruit, we have moved to tractors. And so we had a mechanical revolution, we had a biological revolution, we had a chemical revolution. We started putting a lot more bought inputs on this landscape after the Second World War. But the landscape had already been from the beginning, and this is what I want to emphasize, really all given out to crop production. That's why Iowa has such few public lands and such a lack of diversity in the landscape, as Dr. Jones noted in his keynote. So this process has also created a lot of concentration of farms. And in the last couple of decades, a lot of concentration of livestock. So what you see here is the number of pigs, the increase in the number of pigs, and the increase in the number of layers. In the old days, chickens were not differentiated between broilers and layers. And the disappearance of cattle from Iowa. As Dr. Jones noted, that had consequences in terms of some cropland, right, extensification process. And I want to show you our neighbor to the east. This is the livestock scenario in Illinois. So don't let anybody tell you that this was inevitable in Iowa. As Dr. Jones said, this was a pretty deliberate policy choice on our part. They did not get invaded by pigs in Illinois. So what has been happening to conservation throughout this whole process? Well, as I said, conservation happens when we are in need of taking some land out of production, when environmental problems become particularly egregious. But we don't track it very well. If you look at the history of the census, we are magnificent from the beginning at tracking things like yields, right? To show our obsession with yields and area cultivated, but not really conservation. So there were, in the 1950s, conservation was tracked in the census. Again, those were the lean years of the low exports. But the numbers were not necessarily very good. We don't have a very good handle, for example, even on the loss of wetlands. This is something that I'm working on right now for my next my next publication. And this is the epitome, if you want, of the problems we face. So the Conservation Reserve Program is the daughter of the Soil Bank, the granddaughter of the ACP. We've had it since the 1985 Farm Bill. And what you see here is through the Farm Bills, the caps on the line at the top, the cap on acreage, and then the actual enrollment. And what we see is that CRP caps and CRP enrollment goes down when crop prices are high. So CRP is a dual goal policy because the goal is not just to retire land from production, but by doing so, reducing acreage and increasing crop prices. What we need to do is really rethink this approach to conservation that cedes conservation to the when the push comes to shove to the interests of production and unfortunately the way we have it set up, farm income. Because if we don't do that, we end up where we are, and Dr. Jones made this very, very clear. We're not making progress when it comes to the nutrient reduction strategy at all. So we're going the wrong direction. So these are my parting words for you. Some conservation policy best management practices, okay? We have those two. Conservation policy should have its own goal and should be protected as an area that the public has a say on, regardless of crop prices. We should do better than we're doing now with the nutrient reduction strategy than just counting practices. We should do better at doing watershed level analysis, and we should do way better than the nutrient reduction strategy is doing, because they are completely ignoring the CAFO problem. It's a 300-pound gorilla in the room that nobody wants to talk about. And that's particularly important because as we see these increases of animals on the landscape, we can't just count BMPs, the other kind of BMPs, right? The ones on cropland, as if things were not changing and getting worse when it comes to livestock production and the pollution that that production generates. This will cause me no end to irritation until I die, okay? I really don't care that we have 3 more wetlands when we have twice as many pigs as we did in the 70s. 3 more wetlands aren't gonna cut it. We should also consider the costs and scalability of the policies. How much does it cost to put all the bioreactors that we would need to address the problems that we face? And do farmers actually want to put in those bioreactors? This kind of research is unfortunately missing from our current approach. We don't talk about scalability. And we should also consider the fact, as I said, that now we have these subsidies that are considered given via the crop insurance program. And so if we're giving farmers money, we should consider whether that money is maybe causing some of the problems that then we give money to farmers to solve. That's not very efficient policy. And it's not very effective, okay? Maybe let's start thinking about things like the ethanol mandate and how to do cross-compliance with conservation, with the crop insurance subsidies. Last but not least, public policies should not subsidize activities that only have private benefits. Tile drainage does not have any public benefits. And so when I hear that people talk about tile drainage and help farmers with tile drainage, that is abysmally terrible policy. There is absolutely no rationale for us to do that. Farmers have been putting more tile on the landscape. Over 1.5 million acres of new tile has gone in between 2012 and 2017. We certainly don't need to pay them to do that. They reap those benefits. And in fact, we reap some of the effects of those kind of practices. So I am done in 19 minutes, and I'm happy to take questions if people have any. Thank you.
Thank you, Sylvia. There are a couple of questions. I'll start with Lee. What practical recommendations do you have for us as we lobby the Iowa senators and Congress people for the 2023 Farm Bill?
So I would say there's a couple of things. One is clearly the industry is ramping up protection of the ethanol mandate and the RFS because it expires in 2022. The renewable fuel standard has failed. It has failed as a policy because cellulosic ethanol has never become what it was supposed to become, and the electric cars are going to take over way faster than we planned. So to keep putting money in the ethanol mandate and pushing for more ethanol, therefore more corn, is a losing rural development strategy. I know they don't want to hear it. I know they don't want to hear it, but it's coming anyway. And if we don't get ahead of the curve, Iowa is going to lose more population, and we're not going to see any rural development in any meaningful way. So not renewing the RFS is one thing, and the second thing is let's think about cross compliance. If farmers want subsidized crop insurance, they shouldn't be farming on the 2-year floodplain. If farmers want subsidized crop insurance, they shouldn't be doing fall applications. All those things that Dr. Jones talked about, we are giving these people massive amounts of money. Let's get something back for it.
Thank you. And then Matt has just as a general comment on, thank you for the great historical and social report. Realistic and helpful for seeing us how things are generally. So I have a question kind of on like the follow-up of what you just said on the renewable fuel standard. So what can be done to change the narrative? Because right now we have a lot of people who talk very loudly about a thing and they get very unhappy if you say anything counter that. So how can we change that narrative?
Well, first of all, Ben, thank you so much for being part of this. You know, I think this was a very, this has been a very good day to think honestly about what is happening. And so that's the thing. Let's be honest. I think that the self-censorship that we see from a lot of people, from my alma mater, Iowa State, for example, from a lot of people in positions of leadership in the state on conservation issues, everybody is afraid to say what everybody knows and says behind closed door. So I think what we need to do is be honest to start with, and just, you know, my job, you know, I do policy assessment. There is no way that you can argue that the RFS has not failed. There's no way you can say that, because we are not producing cellulosic ethanol. Every year we've had a cellulosic ethanol mandate, the EPA has had to revise it down. At this point, we should be producing more than 10 billion gallons of cellulosic ethanol. We're not even producing one. So, you know, let's face the music. I think that in Iowa, we hear a lot of like generic, like, you know, let's help the farmers. If this doesn't work, we got to try something else. Doing more of the same is not going to change. Brent wants to stir up trouble, as he is wont to. Soil carbon offsets are a really terrible idea, especially in this system. Now, if we want to pay farmers to convert land to pasture or grassland, like the CRP, as Dr. Jones was saying, that's one thing. But 3-year contracts for conservation tillage, that's not permanent change. It's not clear how much carbon conservation tillage sequesters. So also not clear how additional it is if the farmers were doing conservation tillage already. So it's throwing away more money for conventional, large scale conventional farmers. By the way, the farmers who make money off of this are the big farmers, the conventional big farmers, who are also the big polluters and the ones who use a lot of fossil fuels. So if we can think about different practices, permanent practices, additional practices, practices that favor small farmers and farmers of color, I'm all for it. But not this. This is just like money for nothing. Throw money away, and we can't afford it. The restoration of prairie for, yes, prairie restoration is definitely something that I would support because it is permanent and it is additional, right? And we know that there are benefits in terms of carbon sequestration. But sorry, if I have to hear, I've been hearing about conservation, tillage, and carbon for 20 years. And let me tell you, it really is not a very effective strategy. And it's easy to cheat too on that.
Thank you, Sylvia. This has been a great eye-opener for me to see those stats and to look at those charts and see how it all works. I really appreciate it and hope you'll come back and visit with us again soon. We have so much to learn and so many ways to deal with the system. And as a farmer myself, I'm going, what can we do to, and Lee asked it in the best way, because he's involved in the political side of it and in what we can do for the 2023 Farm Bill. I really appreciate all of you coming. If you and the rest of our speakers can share whatever links and emails, that would be wonderful. Ben, do you have any last words here?
Let's just a few other comments of thanks from our speaker or to our speakers. For our speakers, if you could get us a PDF of your presentation, that would be wonderful. We do now have a new website, so we are going to try to utilize that. And like Chris said, if you can get us your emails, it's great. And I have those, but not everybody who's here might not have them. And then also, again, just one last plug. A lot of the three of the talks you did here today did come from RRWA memberships and donations to our research fund. So thank you for your donations in the past. That has been very helpful to our speakers. And as we had a little bit, we were a little pressed on time. If anybody has any questions for past speakers, feel free to put it in the chat. I think we're done more or less for today though. So unless people have questions they want to ask, I would like to, as Chris said, thank you to the speakers for giving up some of your time and talking about these great things. And then for the rest of you for listening in on a nice but windy Saturday.
Thank you all for being here. I'm going to leave the chat open for a few minutes. And yes, and thank you all for to our speakers, to the RRWA board for helping host, and to all of you for attending on a Saturday morning. I really appreciate it. I put our link, our raccoonriver.org, our new website. And if you're on Facebook, check out the Life in the Raccoon group. We'd love it if you were all members, and the Life in Raccoon River Watershed as a page to share, and we share information as we come. We'll be posting pieces of this presentation and on both of those places as well as on YouTube. So hope to see you all soon. Hope you were that you were able to find something of interest this morning. And if you have ideas for programs and presentations and would like to be members of the Watershed Association, we would appreciate your visit. Thank you so much.
You guys, Chris and Ben, you guys need to have a glass of wine this evening.
Yeah, this was excellent, guys. Thank you so much.
Thanks, Lee.
Chris and
Thanks.
Sylvia.
Yeah,
Good
thanks,
stuff.
Sylvia.
Good stuff.
Yeah.
Wow. Yeah, and thanks to Peter for the research in our watershed.
And Paul too. Interesting cattails that you see everywhere, and I hadn't thought about it at all.
Yeah.
Okay, well, I'm going to put my boat in the Mississippi River to go fishing.
Well, good for you, Chris. Great to see you. We follow that blog with great interest over here in central Iowa.
Chris,
Okay.
do you eat the fish, Chris?
Oh yeah, I eat some of them.
Okay, I'm just checking. Okay.
And I'll put out a blog or a shout out to Paul's blog as well. He has some really good information on there as well. It's very wetland focused. So if you do get a chance to check out his blog, it's also I got some really good stuff.
Thanks, Ben. Appreciate that.
Right. Thank you all. Have a good day.
Yes, thank you all we really appreciated it.
Thanks.
Thanks, guys. Thanks for live tweeting too, Ben.
Welcome. I tried to keep up. You were going through some pretty quick stuff. But yeah,
I probably was as bad as Chris. Man, he was also hard to live tweet about. So
yeah, so I tried to keep up and take notes with everybody. So I hope to have captured the general essence of most of your guys' talks, but we really appreciated it.
Ben, just out of interest, where are you tweeting?
At Maas, Ben Maas.
Oh, yours. Okay. All right.
Yes.
Thanks. Yeah. If you know
so
Andrea,
it was.
sorry,
ahead.
I was just gonna say, if you know Andrea France, just say hello from me. We went through the same program at Iowa State.
I will do that. I don't run into her too much because of COVID times, but periodically I do. So
All right.
she's still doing really good work up there.
Yes.
All right, I'm going to send you the PowerPoint, Ben. Watch your.
All right. All right. Thanks, Paul. I don't see any other.
I'm happy to send you the PowerPoint too, guys, if you want them.
Yeah, either that or just the PDF will be fine, because sometimes the PowerPoints can get a little unwieldy for getting it through the email system. So
I will do that and send it to you, Ben.
All right, excellent. Thanks again, Paul. I'm glad that things worked out. We were able to get you here.
Hey, Ben, I see two Dr. Ben Maas's. Which are you? Are you Maas Ben Maas?
Yes.
Ben Maas, or are you Dr. Ben Maas?
Yes, I have a maroon and gold face mask on in mine.
Got it. All right. Thank you.
All right. Yeah. Thanks, Chris, for setting this up. I think it went okay with the Zoom sign in. So I think it went pretty smoothly as well as we could have expected on that.
I think it did too. I'm glad that it did. So I think we're down to, I don't remember how many. We're down to 13 people. Anybody else have anything for anybody?
Chris, just one thing. I'm looking for research on flood mitigation with cover cropping. If anybody knows about research on that, I want to try to talk with the city folks at Ankeny and Des Moines both on that topic. And so if you, Sylvia or Ben or anybody knows anything about that, send me some research titles if you have them. Thank you.
You put that in the chat too, didn't you, Lee?
I did. Thank
Okay,
you.
because I'm saving the chat and we'll go back through and see what we have. And the other thing we will do is be sharing this back with our participants and our speakers so that we do a follow-up.
Right. Thanks. I'm trying to build an urban rural coalition here in our 4 Mile Creek watershed, and that would be helpful. Thank you.
That's great. Thanks.
I think it might be safe to call it, Chris.
Okay, I see that we have a couple of people from our board still here. I really appreciate all your help and support. And if there is anything that you might suggest for later or speakers and so forth, we'll catch up at our next meeting in April, or if you need to be in touch with me, admin@raccoonriver.org. And I'm sure that you have my chris.henning.50059 if you need to catch me directly.
Yep.
Okay, thank you all.
Chris and Ben and everybody was wonderful. If anyone has any content or anything that they want added to the website, get it over to me and I'll try to get it on there. I'm hoping to get some of the partners added as well. So I think some of these different speakers will help me do that as far as using some of their blogs and things like that for different pages.
That sounds great. We'll be getting you some of this video. Christine left early with her little dog. Well, maybe not. Maybe she is still here.
I think she just left it up.
Okay, maybe she did just leave it up. But in any case, we'll be getting you the videos, and she's going to do some editing so that we have it in snippets for the website. And we'll get that to you soon.
All right, perfect. Everyone have a great day.
All right,
Thank
thanks so
you.
much. Okay.
All
See
right.
you again.
I'll see you later, Chris. I'm glad this went well. And we got about 50 people here. I think the high I saw was 51. So
Yeah.
that's good. I was a little concerned when we were 3 minutes from start and we had like 20. I was like,
I know, I was too.
it picked up. So I'm happy with this, and hopefully it will help get folks with us.
All righty.
All right, I'm going to sign off. Thank you again, Chris.
Yep, I'm going to end the meeting. There we
All right.
go.
I'll see you later. Have a good weekend.
You too. Thanks.